All right, thank you very much. So indeed, we're switching to a little bit of a different big data challenge, also with many species compared to the previous talk. You heard a beautiful metagenomics lecture last night from Curtis Huttenhauer. So before I even start, I wanted to, if I manage to switch this. There we go. Give credit where credit is due. It's actually Jonathan Magazine, my graduate student from my previous job at UC Santa Cruz, who through his passion and his for data sharing, incidentally, and his interest in marine biology, has really started us on a completely new line of project here, which I get to tell you about today. And of course, he's a grad student, so he did all the work, right? We're clear on that. So marine metagenomics, in a nutshell, is the ocean microbiome, the coastal waters samples that we're going to use. Perhaps exemplified best by the early pioneer work. This was predating the famous GOS, Global Ocean Sampling Project. And the other very recent flagship effort is the Tara Oceans, which will actually put about up to 35,000 actually new samples in our databases. It's also a very nice topic for data sharing because you had a lot of, a few privacy constraints when you want to start playing with data. But we're, by contrast to community profiling, just surveying, we were actually interested in the discovery aspect of this field. And so I just used here one almost random paper that I like to exemplify this, is that discovery just being something like seeing something that is odd, that is unexpected, right? And using it, for example, in biotechnology, you could imagine that you could home in on certain species that you find. And in this case, actually, this group from San Diego discovered an association with antibiotic resistance somewhere with corals that had never seen antibiotics, so in so-called pristine environments. Another example that's not marine is that, they found first reads of Plasmodium and Toxoplasma, for example, in Egyptian mummy metagenomes. So a lot of things to discover here, and so I'll structure my talk in the talk that is the majority of the paper that I'm going to discuss, which is a quantification of the obvious intuitive improvement that we would get if we start assembling the reads of data across data sets, which we call pooling in this sense. I'll have to perhaps remind those who are not experts, it's that metagenomics, knowing which species a certain read comes from is not is part of the annotation. You don't know that. You may have millions of species in just a milliliter of seawater. Secondly, necessarily when you start assembling across data sets, you'll have so-called chimera, like puzzle pieces of genomes put together that are not naturally in a genome. And from a biodiversity interest, that is actually a very dangerous thing to do. So that is exactly what we're going to analyze. And thirdly, to know what we're doing, we'll need to simulate some data. The second part is what I'm really terribly excited about, is to think of it as an application of pooled assembly. And that is largely unpublished, so if I have to be very fast at the end due to time, don't hesitate to ask me more technical details. And so our 10 data sets that we simulated, we wanted to reflect the differences in species abundance and diversity as best as possible. And so luckily in metagenomics, there are a couple of popular programs, particularly MetaSim, to which you give a taxonomic profile. So the composition, the species composition that you want to simulate. And then the error model that we used in this case was for 454 reads, because in marine metagenomics, that was up until very, very recently, practically throughout the publicly available data sets were 454 reads. And so what we found in a nutshell, we also used, we did a species survey with a very reasonably permissive species annotation protocol that we found sometimes used in papers. And we used the Marine RefSeq, a specialized database. And what you see here in this graph is actually those 10 data sets are color-coded. And these 56 species, which altogether make up of almost 100% of these entire data, you see here for each species, what percentage of data of the data set, each data point here was represented. So the bottom line, the take-home message here is really we do have quite some ubiquitous prokaryotes. It was an observation that about 75% of the species occurred in at least 8 of the 10 data sets, right? So not in the same abundance, but they did occur. So that's actually a little bit already surprising. We also found that we want to be very careful about species calling because we found a lot of false positives with these simple methods. So with these simulated data sets in hand, what we did was we evaluated and tried to quantify what kind of chimerism we found, and we found only chimerism above the species level. We used the NCBI definition for that. Only a very limited amount of Contigs actually had chimerism above species level, and these are plotted here. And we use Shannon entropy displays to actually quantify this a little bit. So the black line is equivalent to about one foreign read per about in a Contig with 100 reads, and the yellow line is about the same with the 2 different taxa. And so what we found is, and other people had found it as well, one doesn't really, with standard assembly overlap layout consensus assembly Nubler 4454 reads, actually we don't have to worry too much that we go above species, okay, with this. Now the interesting thing for us was that when we here map chimerism of those who were not mixing species at the strain level, we did accomplish what we wanted to, because we wanted chimerism in order to stand a chance of improving annotation. We also developed a more conservative species caller, which allows us later to work with real data, because all of the species calls that we make are pretty much going to be correct. Okay, but before we do that, we have to remember that most of the species in our samples are unknown, right? So we at least made an attempt to model this a little bit by figuring out, the odds of correct versus incorrect calls, independence of the prior probability of our species actually being marine ref-seq. We used the decoy-based approach for that, and so the raw reads are unassembled data, and the raw lines, the gray lines, and the black lines that you see now is intraset assembly. So you see there is an improvement overall, and when we pool, we see like a line just going through the middle. These are 6 data sets that we used, and we also did a leave one out evaluation. What this means is that in this case, for the correct incorrect, of course, pooling doesn't really make a difference. Assembly makes a huge difference. What perhaps also is expected, that we have an approximate correlation with the N50 Contig size, so the amount of assembled reads pretty much. However, also notice that there is quite a little bit of variation between the sets, so that's always to have been kept in mind. And so the next step, indeed, with that conservative species color was to work with real data. We used 42 data sets. We even had some from different PIs to make sure that would work. And we looked at species annotation, but we also extended to look at protein family annotation using Panther and PFAM. And so what we see here in this visualization, the take-home message is at the bottom, right? On average, we did see a lot of improvement, 15% improvement, increase in species calls, and since they're practically always correct, we feel that is quite a good thing. The bar here is perhaps the most interesting part, is the light blue part, which is the additional species calls that were made when we assembled, and the top bar is always the intraset, and the lower bar is the pooled. So we did get the improvement. You can see the details in the paper, but also you see some variation. When we used Panther here as an example, and that show the PFAM data, to exemplify protein function annotation in a way by a hidden Markov model assignment procedure, we see that mirrored a little bit. So what is well annotatable is well annotatable on both ends, but also we noticed that indeed our purpose that we were improving even further, so that these homology-based approaches actually really also benefit very strongly, and also largely a correlation with N50, but not completely. Okay, so this is where things become really exciting now, because now I asked Jonathan, of course, to look at individual contigs and see how, here you see these colors are the 12 data sets. Let me see whether I can point this here. So these 12 data sets contributed to this contig that is represented here with the protein coding genes annotated here that are predicted, okay? So immediately we see that from different regions, from different geographic regions, you have contributions here. And perhaps that's not so surprising because in that first figure with the lots of little colored points, we talked about that there are ubiquitous species out there, right? So not so surprising. What is surprising about this? This was completely an unknown species. So when you blasted it, there was nothing in RefSeq. You couldn't figure out what the species is. Also no close calls. And Jonathan told me I have plenty of Contigs like that. We have lots of contributions. They're obviously ubiquitous, but they are uncharacterized at the species level or unpredictable at the species level. So he got quite interested in this, and so the circle is where his the Contig that you got is located if you do a principal component analysis on what we call a geographic profile, which is nothing, is extremely simple. It's just a proportion of reads that come from different geographic regions, right? And you see how these clusters are forming? So he got very excited by that, and as life goes, immediately he got scooped, which means his prediction that the other contigs in this cluster, if every circle is a contig, that would likely also belong to the same genome, perhaps because they have similar geographic profiles, was validated by the fact that this was a prokaryote that was a gamma proteobacteria that was sequenced by a single-cell genome sequencing project where they were targeting ubiquitous species. So he got scooped in that sense, but also came closer to his PhD because this is a proof, right? So we saw also that here some of the, if he blasted, he also found a lower level blast match, and he saw that these circles here, the red circles, actually they are in a different cluster. So what we're presuming is that we're separating this species and its cousin, so to speak, right? And we can prove that by showing that there are overlapping regions of genome that are distinct from one another and show that phylogenetically. So the bottom line is we can actually, in many cases, seem to be able to separate groups of contigs from the same bug. And so this are overall 1,700 and something contigs of that greater than 1 kb. So now, of course, the Odyssey began to look at different clusters, right? And he got scooped once again, in a way. This one here, the Rosiobacter, and long story short, we ended up with E and F, where the similarity to known species is actually at a level where you could argue that that is probably a new discovery, right? What he was looking for. And E and F also happen to be have contributions from a San Diego sample. That first peer that you saw on the slide, that's actually San Diego where Jonathan got to do a PCR validation. So, but first, before that, actually, he also noticed that computationally you can actually find further clues, and one of them was by integrating the Gauss data or using the Gauss data. So these are Sanger reads, which align sometimes to, you just use them exactly like paired end reads in a way. So they bridged by Gauss reads, which again were often from different geographic locations. So here we have his favorite is cluster E, and then cluster F we also looked at. The black lines were the Gauss data. The gray lines that you see are paired end data that you could also overlay. So you get a corroboration, but ultimately we wanted to send him to the lab and really take this home. And the way we did this is simply to have here, you see two contigs from this yellow cluster, right, from the putative Rosiobacter-like thing. And what we simply did is we designed a primer based on the San Diego. We went back to the old sample, right, from which originally that came. And so we're now no longer producing chimeric sequences, but we're producing actual genomic fragments. And we could also, in this case, predict how long these bridges should be, and we get them within like a day or two. So it was actually really, really easy and very satisfying. So what do we know about this bug? Well, not really very much. Functional and computational characterization might be in the works, if you like. At the moment, we only, this just exemplifies the type of things we can do, is of course, phylogenetically, we find it as a sister, somewhere in a sister clade to the Rugeria. But also we see here the Gauss data, where is it? Here. That was indeed from Gulf of Maine, which is yet another geographic region. So it corroborates that it's very ubiquitous, and we can keep doing that. So all very exciting. As I said, in the end, I view these discovery approaches as something that might help us prioritize which of these prokaryotes we should go after to eventually, try to figure out how to culture them. So I'm not saying that we can immediately apply them, but we are definitely hoping that some of these methods and visualizations that we have will be helpful also to others. And we want to especially thank, of course, Brian Pelenik, who lives in that lovely Scripps Institute where Jonathan was, and our friends and colleagues Terry and Jeff and John for helpful discussions. Thank you very much.
Oh, I can already see a question here.
Yeah, thanks for the great talk. I was wondering, what do you think about having the longer reads, like probably PacBio or Nanopore? Would it improve the pooling assembly? And then would you take this trade-off, having the longer reads but high error rate?
It's a good question. We've actually not gone so far yet. We are just in the process of figuring out Illumina, right? As the next step, which is kind of the opposite. And I think there will be challenges every time. I should also mention something that I kind of let slip under the table. We have tried different assembly algorithms, right? And it's not as clear cut, right? As you saw, we didn't do a parameter optimization. So my general feel would be we would have to try. I think actually maybe we would get even longer contigs. I think it depends on what you want to do with it, right? If eventually you are trying to get at biodiversity, then mutations become terribly interesting, and then I would be slightly worried with the error rate, but I'd be happy to learn. Actually, I'm more of a short read person.
Over there is a question.
So great talk. You were using Shannon entropy,
yes,
to identify chimeric
to kind of quantify, to evaluate, right? To kind of express.
Right. So I have two questions there. One is, what was your intuition in using Shannon entropy for that, and why is it working? And the second question is, how are you calculating it?
So that is. So the Shannon entropy, I think, was used at least by one other study as well, who was just sort of probing within intraset and trying to address this question and kind of like thing. And so it is regular Shannon entropy by read.
So is it?
So it's by read with respect to the majority of species. Okay, so it's a contaminating read in the contig.
So is it based on base composition, or is it
on read composition? Read. So reads contributing to this contig. Okay. So what you saw on that axis was actually the number of reads. Mm-hmm. Yes.
Yeah, thank you.